Hello, my name is Madison Lawrence. I am a junior neuroscience major at Hamline University this year. This summer, I conducted research at Old Dominion University within the Frank Reddy Research of Bioelectrics. I wanted to share a little bit about what we found and what I learned. My project was titled The Effects of Neurodifferentiation and Viability by Cold Atmospheric Plasma Treatment on IMR-32 cells. Essentially, the problem that we wanted to research was regeneration, due to the fact that when the central nervous system or spinal cord is damaged, it is very difficult to have regeneration and full function after an injury of this caliber. Or if you have cancer or a pathogen that causes this damage, it is very difficult to target these cells without damaging the healthy ones. Evidence suggests that with cold atmospheric plasma or plasma activated solution treatments, we could do just that, eliminate the harmful pathogens or cancer cells without killing our healthy cells. The cell line that I mentioned, IMR-32, is a neuronal neuroblastoma cell line, and it is a good model for differentiation as it has similar projections to the cerebral cortex. We measured the outgrowth and using IMR-32 software, and that is how we came up with the averages and measurements of this study. Retinoic acid with differentiation medium has been proven to be a successful treatment for this regeneration as well, which is why we decided to use it as our control. The culture conditions of this study was using Eagle's minimum essential medium with fetal bovine serum and penicillin streptomycin. Our coating surfaces were PBS with laminine or fibronectin. We eventually narrowed down to a coating surface of laminine strictly and a density of around 8.0 times 10 to the 4th cells per well. The PAS treatment involved a surface micro discharge air plasma device, and the PBS was exposed to this plasma for 45 seconds, and the PAS was used directly to treat the cell cultures. For differentiation, we used a neurobasal medium with L-glutamine, B27, and PS as well. Once again, the control was with this differentiation medium with retinoic acid. For proliferation, we used an MTT incubation, and the viability was calculated with net absorbance of the treated sample divided by net absorbance of the control. And we also did a fluorescent staining assay to further look at the differentiation and the outgrowth more clearly. The imaging of the fluorescent stained cultures was using a fluorescence microscope with a CellSense software, and the unstained was with a compound microscope and infinity camera, as well as Luminera software. Our results narrowed down the coating surface, as I mentioned, to laminine, and this was because the morphology and differentiation was better than the fibronectin for our specific study. The image comparisons also indicated that the PAS is comparable to the RA control that I mentioned. We also found the viability of the PAS treated cells to be around 100%, which was again done by the proliferation assay. And with IMR-32, we measured individual outgrowths of each sample and averaged them. We found that the PAS was statistically significantly different than the sham control, which was our no treated group. And there was very little difference to no difference to the RA treated cells and the PAS cells, which was overall good evidence that the PAS could be a potential clinical treatment. In the future, we could enhance the study, of course, with more time and maybe with measuring our reactive oxygen species to narrow down the exposure times or different measurements. And another assay we could perform is looking at glutamate isotoxicity. But overall, our study was very successful, and I am very grateful that I was able to conduct this research. Thanks for listening.